wonderful to be back here because many years ago, I was an alumni of SAFS. So it's a pleasure to be back here, and thank you so much for having me. Today, I'm going to be talking about some work that I conducted prior to my current role at NOAA, so a little less fisheries oriented here. This is some work I did on the evolution of fish diversity. And I think when many people see diversity in this context, what they immediately think of is species diversity. But I want to emphasize that diversity can mean a whole lot of different things. And when I was trying to figure out how to start this talk, because starting a talk is usually the hardest part, I typed the title of this talk into one of those AI image generators on a whim, and it actually sparked something for me. Let's see if this will work. So this is the image that was produced. And I think at first glance, you might think it captures diversity, but I want to emphasize that it kind of doesn't, especially when you're talking about the scope of all fish diversity together. And aside from some of the really hilarious misses with a double-headed fish and fish with 4 pelvic fins, you could squint and say maybe it captures some species diversity, maybe some color diversity, but I would argue that all these fish look pretty much the same in terms of body shape, in terms of fin placement, whatever number of fins it does have. And they're all kind of in the mid-water swimming around. And so I think this really falls flat and totally misses the mark, because when we're talking about fish diversity as a whole, it's incredibly diverse. Fishes have maximized and exhibit extensive variation on basically any axis that you can imagine. So if you're talking about morphology, ecology, physiology, function, behavior, etc., fishes are incredibly diverse. And so the purpose of my work and my research program is to understand the mechanisms that are driving this diversity across fishes. And regardless of the type of the diversity, one of the most unifying features is that this diversity is unequally distributed. So what do I mean by that? Well, so for example, let's take the family Cichlidae. Across fishes, there's about 1,700 species, so incredible species diversity, yes. But there's also incredible other types of diversity. So there's a lot of body shape variation, a lot of ecological variation. And then there's other clades of fishes that only have maybe a handful of species, and it would take a taxonomic expert to tell them apart. So what does this unequal distribution of diversity mean? Well, it hints that there's variation in the underlying tempo and mode of evolution. That is, the dynamics of evolution itself differ across clades, but also across spatial scales and temporal scales. And specifically, comparative methods allow us to harness our knowledge of evolutionary dynamics and connect it with finer scale ecological trends to explore kind of broad scale patterns across the fish tree of life. So all of my work is focused on leveraging our knowledge of evolutionary history using this comparative framework to understand the mechanisms that are driving patterns of diversity. So in that vein, I'm going to present 3 different vignettes of my work today. And these are all different vignettes that are focusing on one particular ecological feature, which is habitat. But I'm going to be looking at it through a few different lenses. So the first part of my talk focuses on how habitat can influence morphological diversity across fishes. So I'm going to be looking at trends in body shape evolution. And then the last part of my talk focuses more on that traditional species diversity, but on a global scale. So first, as I mentioned, diving into morphological diversity. And specifically, I'm looking at how the benthopelagic axis can influence and shape patterns of morphological diversity. Now, as a fisheries department, I imagine a lot of you are very familiar with the benthopelagic axis, but just to refamiliarize you about what it is like to be a fish living in these 2 different habitats, here's the pelagic habitat. Looks very fun, looks exciting, huh? There's not much to do there if you're a fish. You can swim around, but there's really just water on all sides. There's nothing to interact with. There's no complexity to interact with. Whereas benthic habitats are incredibly diverse. So you see habitats as varied as coral reefs and hydrothermal vents and intertidal zones. And these habitats are all incredibly structurally complex. And that promotes ecological opportunity to diversify for different fishes. So the opportunity to diversify is potentially much greater in benthic habitats than just good old pelagic habitats. And no shade to those of you that focus on pelagic habitats, but it works for my story here, so go with it, please. And we see, in an effort to minimize drag, extensive convergence on streamlined body shapes in pelagic environments, especially in high-performance fishes like tuna that are exposed to a lot of drag swimming through the water. But for benthic fishes, that is those fishes that are living in contact with the benthos, they might look wildly different. There's no selection on them to necessarily minimize drag to the same degree. So it's unclear what the axes of morphological variation would be in benthic fishes. But of course, we all know that fishes don't neatly fit into these two binary categories, and there's a lot of fishes that have some degree of interaction with both habitats. And these demersal fishes are potentially under very different selective pressures, having to both swim constantly and maneuver around structures on the benthos. And it's important to note that species, well, across fish evolution, they have radiated into these three habitats numerous times. So it's kind of this perfect study to look at the evolutionary consequences of this axis using fish. And we know through a lot of other studies that have looked at this axis that there are specific morphological patterns associated with these three habitats. And these are recapitulated at both the intra and interspecific scales. So for example, good old classic stickleback, I know this would connect with people here. You see pelagic forms that tend to be much more streamlined, and you see demersal forms that tend to have a deeper body shape. And we see this, as I mentioned, recapitulated again, but at higher taxonomic levels too. So this was a study looking at grunts, or the Haemulid family, and they found yet again that the pelagic-oriented species are more streamlined, the demersal-oriented species are more deep-bodied. Okay, great. So we know what each of these habitats, what selective pressures it has on body shape. But we don't, because these studies so far really haven't looked at the benthic axis. What they've studied is much more of a pelagic demersal axis, and they've totally neglected benthic fishes altogether. So we have no idea what the entirety of this axis is doing for fish morphological evolution. And certainly these studies have not been on the sort of a macroevolutionary scale that I'm proposing here. So the first part of this talk is going to be asking two different questions. I want to look at how habitat influences body shape diversity across fishes. And then the second part looks at how habitat affects the rate of morphological evolution. So that's the speed at which these clades are accumulating body shape or morphological diversity. So to do this, I've collected with a huge team of people. This is by no means just me. It was a massive effort over multiple years. But we collected 8 linear measurements of body shape on fishes that were preserved at the Smithsonian. And these linear measurements are designed to capture kind of general functionally important axes or traits from fishes. And we've matched this to a phylogeny that was previously published. And with this combination, we were able to get information, both phylogenetic and morphological information, on over 6,000 species. And so each one of these blue lines here is a species that we have both sets of data for. And this data set is really cool. It's really powerful because it allows us these insights into with a resolution across millions of years that's just not previously been accessible without a data set of this size. So with that being said, I have cut this data set in half. So this first part of my talk is only going to focus on marine fishes. And so I'm working with a measly 3,300 species here only, but that's still enough to look at some really cool trends. So I've taken each of these 3,300 species and I've broken them into these 3 habitat categories. If you haven't gotten it yet, I'm going to be keeping these colors consistent throughout the talk, or this portion of the talk. So pelagic will always be light blue, demersal dark blue, benthic orange. And as I mentioned, pelagic fishes are going to be those that spend 95% of their time just in the water column. Benthic fishes likewise spend 95% of their time on the benthos, physically in contact with the substrate. And then demersal fishes have some degree of variation between those two. So we've taken those 8 morphological traits, log transformed and size corrected them. This is just a process to kind of control for allometry and to control for size variation across species. And then we've matched everything to the phylogeny, so took out all the species that we did not have information on. So without further ado, let's get to some data. So firstly, what I'm showing you here is that elongation axis. And over again, this is described as the primary axis of variation across fishes. Basically, if you get a pool of fish together, elongation is going to be that maximum dimension of diversity. And when we look here, I have the curves for the 3 different habitats. The most important takeaway is I want to draw your attention to this region of the space over here, which represents some convergence between pelagic and benthic fishes on these extremely elongate forms. And so here's some examples of what some of those fish in the different habitats look like. And an elongate body shape is really interesting because it offers specific functional advantages. So an elongate body is really useful for steady swimming performance, which is pretty useful if you're a pelagic fish. But it also offers enhanced maneuverability and flexibility, which is also useful if you're a benthic fish. So it makes sense that we're seeing convergence on these shapes. And furthermore, it's a really developmentally straightforward way to just change your body shape. You can just add vertebra or elongate the vertebra you already have. So this is a pretty interesting insight. But I want to move to one of the other primary dimensions of body shape evolution across this data set, and that is this body depth, body width axis. So here I'm showing you body depth increasing to the right and body width increasing to the left. And the interesting thing here is I'm just showing you demersal and pelagic fishes, and despite our expectations at the start, and despite what all these other studies have shown, on average, demersal fishes really aren't deeper bodied than pelagic fishes. So this kind of unhinges our longstanding theory that demersal fishes are more deep bodied. And what's more interesting is when we actually incorporate those benthic fishes that, again, have not been looked at before in this regard, at least, we see something really interesting here where benthic fishes are occupying regions of space, namely this really flattened, wide-bodied shape that we're just not seeing other fishes able to get to. So they're doing something completely different as soon as their body becomes in contact with the substrate. Now, if you're observant, you might be noticing the secondary peak over here, and you might be wondering, what is going on? And this is our flatfishes, which are totally doing something different. So instead of maximizing that body width axis, what they're doing is functionally mimicking having a wide body, but they have an incredibly deep body and they're laying on their side on the benthos. So they're doing basically the same thing, but in their own little flatfishy way. So we know now on average that there's not much of a difference between pelagic and demersal fishes, but there is a big difference between either of these two categories and benthic fishes. So now let's throw all of our data back in, because I've only been talking about a few dimensions of body shape, but we have all 8 of those morphological traits to play around with. So I threw them all into a PCA together, and PC1 is body length, PC2 is body width. Each point here is a single species. And just to orient you to this space, we have our eels to the far right, very elongate, makes sense. As I mentioned, we have our really deep-bodied flatfishes over here. And then at the top, we have our anglerfishes, which are notoriously very wide-bodied and dorsoventrally flattened. But what's really interesting is when we look at the entirety of the space occupied by each of these habitats, we're finding way more morphological disparity in benthic fishes. And this implies or suggests at differences in the dynamics of evolution in benthic fishes. So not only are their body shapes different, but this suggests that there's faster rates of morphological evolution as well underlying this. But to actually evaluate it, what we need to do is go back and incorporate that phylogeny. So here what I'm going to be doing is a method called stochastic character mapping. And essentially what this allows us to do is, given that we know the habitat occupied by every single one of the species around the tip here, that's what I'm showing you, we can reconstruct the evolution of habitat through time. And this allows us to do something like this. And we could do this a whole bunch of different times, reconstruct that evolutionary history, and that allows us to iterate over variation in the timing and the positioning of those transitions. So I've done this 100 different times, and one of the main takeaways, number one, across all the stochastic character maps is that there's really high phylogenetic signal to habitat. So what you can see is that the colors are really clumping together on the phylogeny, so closely related species tend to be in similar habitats. But what's really fun is with a data set like this, with 100 different sim maps is what we call these, what we can do is reconstruct habitat transitions and look at the rate of transitioning to different habitats through time. So the arrow weight here corresponds to faster rates or faster number of transitions between the two states. And so what we can see is that demersal and pelagic fishes are kind of bouncing back and forth between those habitat states through evolutionary history. We see a lot of fish able to move readily between those two habitats. But what we also see is very few transitions to the benthic habitat, which implies that it's really evolutionarily difficult to become a benthic fish. We can also use these sim maps to model trait evolution. We can model trait evolution and determine the best fit model of morphological evolution using these sim maps. And universally, what we're finding is that the best fit model is a multi-rate Brownian motion model, which implies that there's different rates associated with the different habitats. And I'm just going to show you each of the primary dimensions of body shape, the results, but it's basically consistent regardless of which one of the 8 traits we're looking at. The rates of morphological evolution are universally faster in benthic fishes. You're seeing them fall out to the far right every single time. In fact, the rates of evolution are almost 1.6 times faster than that of pelagic and demersal fish. So in summary, what we're finding is that on average, fishes look really similar, but disparity overall varies quite a bit with habitat. And the degree of interaction with the benthos in particular can have some really broad morphological implications. That is, they're able to achieve these body shapes that other fish just aren't able to get to. So overall, it implies that it's really difficult to become a benthic fish, but the opportunities to diversify once you're actually there are way greater. So benthic habitats are providing a lot more ecological opportunity to diversify in body shape, but also, importantly, there's not as much selection from drag to create that streamlined body shape. So it's freeing them to explore new areas of morphospace. And more generally, what this study is showing is that habitat colonization can have some really broad consequences for fish evolution and diversification. But marine environments are only half that story. Remember, I told you at the start that we cut our data set in half. So let's bring back in those freshwater fishes and see what's going on there. And when we talk about marine and freshwater fishes, when we're just looking at the surface area of the Earth, marine environments are around 70% of Earth's surface area, whereas freshwater environments are around 2.5% of Earth's surface area. But if you're talking about fish diversity as a whole, that is the number of species diversity, they're basically equal between freshwater and marine habitats. So there's something going on here, and this implies that there's an interesting evolutionary story, which is of course something that I'm going to get into. And when we're zooming in at the interface between freshwater and marine environments, there are these really profoundly different environments for the fish living there. So freshwater environments are really complex, and they're fragmented too, highly fragmented. And marine habitats are really open and interconnected. And the different features can have very significant consequences for fish evolution and diversification. But bringing back the previous story, there's also opportunities to diversify along this benthopelagic axis in both freshwater and marine environments. And we also know that they've done this repeatedly through evolutionary history, and we have particular expectations of what fish in each of these habitats looks like, although that story seems a little more complicated now in light of the previous part of this talk. But we have expectations going into this. So this sets up a really interesting juxtaposition where we find examples of very consistent patterns of morphological evolution associated with particular habitats, and thus theoretically similar selective pressures in each of those habitats, but in two very different ecosystems. And so I'm asking here is if we see similar patterns of diversification across marine and freshwater systems. So I'm using the same data set as previously, still those 8 morphological traits, but I'm breaking them into 6 categories now. So again, keeping the colors consistent, but freshwater is always going to be in these dotted lines. Think of it as these systems are fragmented, and marine habitats are always going to be in the solid lines. So we have freshwater pelagic, freshwater pelagic, marine pelagic, freshwater demersal, marine demersal, etc. And going back to that PCA I showed you already, PC1 is that composite trait that represents morphological diversity across a group. So this is PC1 for all marine fishes, and we already know that's elongation. But here what I'm asking is if species in the same habitat diversify along the same axis. So are their PC1s similar? And if we were to take out just benthic species from this plot on the left, PC1 in just benthic fishes is actually very different than PC1 across all fishes combined. And in fact, if we put that line over here, it's nowhere near parallel to PC1 for all fish together. So now we know that the morphological evolution, or that primary axis of variation in marine benthic fishes, is something like this. And we can do this for every single one of those habitats in marine systems. And now we know the primary axis of morphological variation for each of those habitats. But what we want to know is about corresponding habitats between freshwater and marine systems. So we have our 3 marine PC1s, and we can calculate our 3 freshwater PC1s. And then what we can do is compare corresponding habitats. And I'm going to be doing this across all 8 dimensions. So this is a multi-dimensional analysis taking into account all 8 of those morphological traits. So the PC1s are similar. We can calculate an angle between them. And if they're similar, they're going to have a very small angle between them. That's going to imply there's very similar axes of morphological diversity. But if they're very different, it'll have an incredibly large angle, maximizing, the maximum would be 90 degrees, which would be wholly uncorrelated axes. So let's do this for our corresponding habitats. And here's what we find. These angles are really small. They're actually, this is implying that there's parallel morphological evolution across habitats. This implies that habitats are having very strong and very similar selective pressures on body shape, regardless of whether they're freshwater or marine. And I actually implemented a significance procedure, simulated traits under a NOAA model of morphological evolution, compared these angles, and they're actually statistically smaller than we would expect by chance. So there's something going on here. And we can look at this in another way, because I'm a very visual person. So this is more of a quantitative way to assess it, but I also like a qualitative way. So we can look at the individual trait loadings on PC1. So I'm going to show you a radar plot here, and each one of these spokes is one of those original 8 morphological traits that went into this PCA. And we can plot the absolute value of these loadings here on this radar plot. So the closer each of these points is to the outside of the circle, the higher the loading, the more that trait matters to variation across PC1. So for example, standard length matters a lot to variation across PC1. And we can overlay the corresponding freshwater habitat, and you can really start to pick out the nuanced differences in these traits and how much they're mattering to morphological evolution across each habitat. So maximum fish width, for example, matters a lot more to marine benthic fishes than it matters to freshwater benthic fishes. So here's those results for each of the different habitats. So not only do we have these small angles, but we can actually look at exactly what traits are contributing to those small angles. But if you're like me, this brings up a really interesting second question, which is, what's going on here? What is driving this parallel morphological evolution? So there's 3 possible processes. It could be by chance, but I already kind of ran that procedure of simulating traits under NOAA models of evolution and ruled out that chance, that ain't it. So this leaves 2 possible processes here, which are shared ancestry and adaptive changes. And so to tease these apart, what we can do is assess phylogenetic signal. So if the axes are similar due to some sort of shared ancestry, this would imply that morphological evolution proceeds along with the phylogeny. So closely related species are going to be in similar habitats and have similar morphological axes. And then we would expect to see very strong phylogenetic signal in this case. But if it's due to some sort of adaptive change, we would actually expect to see very weak phylogenetic signal. So this would imply that morphological diversification doesn't align with the phylogeny at all, and closely related species are maybe in very different habitats, and selection is what's driving similar axes of diversity. So to tease these two apart, what I've done is I've implemented this new method called a phylogenetically aligned PCA, or PACA. And so what this is doing, instead of a traditional PCA, which aligns axes to the maximum variation in the data set, what this is doing is looking at aligning components to the maximum phylogenetic signal in that data set. So remember, phylogenetic signal is that tendency for closely related species to resemble each other just due to shared ancestry. So if I were to plot PACA1, it would actually be that linear combination of variables that has the highest phylogenetic signal in the data set. So what we can do is compare PACA1, that phylogenetic signal, to PC1, that morphological signal, within a single habitat. And that tells us how closely aligned morphology and phylogeny are. And we can do this using the same exact procedure, calculate the angle between those vectors. If it's a small angle, that means they're very similar. If it's a large angle, they're very different. And that's exactly what I did. So here I'm showing you the original PC1 in color. You already saw this. And then PACA1 is this gray line below it. And the angle between these two vectors is above each of the radar plots here. And the main takeaway for marine habitats, at least, is that there's very small angles here. Morphological evolution is proceeding along with the phylogeny. The story gets really interesting when we look at freshwater fishes because they're doing something totally different here. These signals aren't aligning at all whatsoever. The angles are much larger. So what this implies is that there's very different processes driving morphological diversification in freshwater and marine fishes. But wait, we previously saw that there's parallel evolution. So there's different processes driving the same pattern of morphological evolution. So we can also calculate phylogenetic signal just purely across all dimensions. And I did do that kind of as a sanity check. And what I found, this is Bloomberg's K, if you're familiar with it. What I found is that the signal, phylogenetic signal, is universally small in freshwater habitats and universally large in marine habitats. So this kind of makes sense with the results I already showed you. And going back to those two processes that could be driving parallel evolution, well, what we find is a really interesting dichotomy where marine clades tend to diversify in a way that is predicted by their phylogenetic relationships, but freshwater clades aren't. So because we're seeing these parallel axes of diversity, we know marine and freshwater fishes are converging on PC1. And that implies that habitat is imposing some very strong selective pressures on body shape evolution. But marine fishes have that strong phylogenetic signal and freshwater fishes don't. They have a completely opposite trends. So what I think is going on here is that it relates back to the biogeographic differences between freshwater and marine ecosystems. So oceans, remember, are these very open and contiguously connected ecosystems, which allows for greater population connectivity in the marine realm. And we're going to see this in this study as a pattern of widespread phylogenetic niche conservatism. This is whereby fishes that are closely related are just going to stay in the same habitat and diversify there. And in contrast, we're seeing virtually no diversification along the axis described by phylogeny and freshwater fishes. And again, I think this is relating back to the biogeographic nature of that environment. So freshwater habitats are these really highly fragmented habitats, even on potentially very small spatial scales. And that can facilitate iterative ecological adaptation, which is exactly what we're seeing here. Basically, freshwater fishes tend to invade new habitats and then just diversify in a similar way in each of those habitats. So overall, what this study is showing is that despite very divergent evolutionary processes, habitat is imposing some very strong selective pressures on body shape overall, which drives very consistent patterns of morphological diversification on a global scale. With that, I'm going to totally switch gears and talk about something completely different, which is species diversity. I feel like we've dove into marine diversity well enough there, and I'm going to pivot and talk about species diversity across depth and latitude on a global scale. This is using a totally different data set, so get the rest of that out of your head for now. And I'm going to drink some water and then we'll launch into it. Okay, so if we're talking about spatial trends, global trends, the biggest spatial trend in terms of diversity is the latitudinal diversity gradients. And so this is the idea that species richness increases towards the tropics and decreases as you move towards the poles. And this pattern has generated a lot of scientific interest over the years because it's basically ubiquitous across marine and freshwater environments and across phylogenetic groups. So we find this pattern in birds, in plants, in mammals, in amphibians, and of course, in fishes, why you all are here. So the causes of this pattern, I would say, are pretty hotly debated in the literature. There's no true consensus across everybody, but I would say one of the leading hypotheses for this pattern is that there's faster speciation rates in the tropics. So this is the number of speciation events or branching events on a tree over time. And if your rate of speciation is faster, that would lead to more species. Makes sense. But while faster speciation rates have been found in the tropics for a lot of different phylogenetic groups, fishes actually exhibit completely the opposite pattern. So for fishes, the fastest rates of speciation are completely biased towards the poles. So that sets up a really interesting dichotomy where the greatest number of species are in the tropics, but the fastest rate at which these species are arising in modern day is in the poles. So this leads to a really interesting spatial mismatch here. And even more interestingly, this trend is driven almost exclusively by 5 clades of fishes, many of which you are probably familiar with being based in the Pacific Northwest. So these are the eel pouts, rockfishes, flatfishes, snailfishes, and then probably less familiar with the Antarctic ice fishes. So the higher latitudes, which is where all the fish evolution action is in modern day, at least on this scale, these environments are also being subjected to the fastest warming rates. So elucidating the mechanisms that are driving rapid diversification in these clades is kind of a pressing issue because it shows us how to maintain biodiversity in the face of a changing environment. And one potential driver of this pattern of rapid speciation rates at the poles is the depth axis. And nobody's looked at this before, but it is this kind of ubiquitous feature of marine systems, this depth gradient. And there's really dramatic shifts in terms of the environment along this gradient. So in the shallow seas, they tend to be relatively well lit, relatively warm, well mixed, very different environment than the deep sea, which is basically this, I don't know, very dark, very cold, high pressure, very difficult environment to live in if you're a fish. And fishes have occupied the entirety of this gradient, basically. So we find fish obviously in the intertidal zone, but there's fish been found all the way down to like 8,000 meters. So reflecting this environmental gradient, and of course the physiological challenges associated with this gradient, we find some broad shifts in the structure of fish communities as we move down through the depth layers. And this suggests that shifts in these environmental features are driving shifts in fish evolution and diversity. But of course, these features, these environmental parameters, despite varying on this depth gradient, they also vary on a latitudinal gradient, such that the environmental parameters tend to be slightly more uniform at the poles than at equatorial or tropical latitudes. So theoretically, they could be driving trends in speciation rates by latitude. But specifically what I'm interested in here is not just the depth gradient, but its depth lability along this depth gradient. So the idea behind this is that invasion of novel habitats at different depths may create ecological opportunity, which would drive diversification. And it would also create, isolate populations vertically, which would limit gene flow and also cause speciation. So let's pretend we have a clade here, clade A, which has high depth lability. This clade will be able to freely disperse along the depth axis, and it might be more likely to colonize novel environments along that depth gradient. And that would lead to repeated local adaptation and rapid speciation. But in contrast, if we have clade B that has very low depth lability, we wouldn't expect it to experience that same increased speciation rate. So if you're plotting speciation rate by depth transition rate or depth lability, what we would expect is some sort of relationship like this. So we know that niche lability, more broadly at least, can influence and drive high diversification rates. This has been found in other organisms and other phylogenetic groups, but this really hasn't been looked at on a depth scale for marine organisms before. And specifically what I want to know is if diversification along that depth gradient has been a key element in the success of high latitude fishes, with a specific focus on these 5 clades here that we already know have those rapid speciation rates and are kind of driving those latitudinal trends in the first place. So I've taken a data set of around 4,000 species of fishes, and I've broken them up into categories by depth and latitude. So for the depth categories, we have kind of broken along traditional boundaries here. So shallow is anywhere from 0 to 200 meters. This is our epipelagic zone, very well lit, relatively warm, and it has the mixing layer. Next, we have the intermediate depths, which would correspond to the mesopelagic zone. So this is where temperature starts to decline pretty quickly, light starts to decline very quickly, and conditions start to shift. And then we have our deep zone, which is completely black, no ambient light, very cold, very homogeneous in terms of the oceanographic abiotic parameters for fishes living there. So those are our 3 depth categories. And then we have our 3 latitude categories. And I've also kind of designed these around typical climatic boundaries and major weather patterns. So we have our tropics between 30 degrees north and south of the equator. We have our temperate zones, which is anywhere between 30 and 60 degrees, and then our polar zones anywhere north of 60 degrees. And I've broken species into these latitude categories based on the latitudinal midpoint of their distribution. So the next thing to do is, of course, reconstruct that history of depth evolution across the phylogeny. And I'm using that same phylogeny here. So this is that Roboski tree. And again, we know at each of these tips, each of these species has a certain depth category. How am I doing on? Okay. Has a certain depth category. And we can, again, paint one iteration of this history of depth evolution on this phylogeny. But we do this way more than one time, and that averages over uncertainty in that timing and the placement of those depth transitions. But I'm not just doing this on a small scale. In fact, I'm doing this across a large phylogeny. And so this gives us really cool insights into the history of depth evolution across fishes as a whole. And next thing I've done is I've simulated traits under a null model of evolution. This is Brownian motion, to determine the expected number of depth transitions by clade. So we know how old clades are. We know the most recent common ancestor is a clade. We know how many depth transitions we see empirically. And now we can simulate and see, okay, how many depth transitions do we actually expect at a clade level, just based on how old that clade is, because older clades are probably going to diversify more on the depth gradient, just given more time to evolve. So I've done this a whole bunch of times, and here's what I found. So here is a list of all of the clades I've looked at. These are just clades that had enough representation in the phylogeny to be able to get an accurate assessment of depth transitions. And those red clades there are, again, those 5 focal clades that have those really fast speciation rates. So here is the depth composition of each of these clades. And I'm going to be filling in the number of transitions. So this is the number of depth transitions. And the closed point is going to be the observed number of depth transitions in that clade. And here is the expected number of transitions based on that null model, with the open point being the median expectation. So the green points are going to represent observed transition rates along that depth gradient that exceed our expectation. And here's what we find. So the first thing I see is that all of our red clades diversify way more along the depth gradient than we expect by chance. So there's something going on there, albeit they're not the only clades that are diversifying along the depth gradient, but our focal clades are all up here towards the top. We see a lot of clades that are kind of boring. They're kind of just within that expectation. And then I really want to draw your attention to the bottom part because I think this is fun. Luke, you'll enjoy this. These, for those of you that are familiar with tropical coral reef fish, these are our damselfishes, our gobies, our wrasses. Basically, if you have been on a coral reef, you have seen at least one of these, and I would wager probably many more. These are the stereotypical groups of fishes that just absolutely dominate coral reefs. They are incredibly evolutionary successful. They've proliferated like crazy, and they are falling out near the bottom of this. So they are doing something else, but the depth axis is not it. So here I'm just showing you latitude and then the number of transitions again. And this is just kind of to underscore that point that these clades are all falling out near the upper right here. So they're high latitude, high speciation rate, high depth transitions right here in these clades. But I always also wanted to look in this and see if there's something else going on here when we look at across everything all together. So not really breaking things down into clades, but looking at the picture as a whole. So what I'm going to show you here is kind of a non-clade-wise approach to look at very similar trends. This is based on an MK model, which is a common model of discrete state evolution. And this allows us to visualize the rate of transitions across both depth and latitude. So I'm just going to show you the tropics to start with. You can see that there's each of the depth categories here. Again, that color is pretty consistent. And there's going to be arrows filling in. And the width of this arrow corresponds to that rate from one state, in this case, deep tropical zones, to another, in this case, deep temperate zones. So what you can see here is there's a relatively small, slow rate of evolution that we're seeing from the tropics to temperate deep zones. And we see a relatively similar rate, a relatively similar width of the arrow from the deep tropics to intermediate tropics. So if we fill this in for all the tropics, these are the depth transitions we see in the tropics across evolutionary history. There's basically not much going on there. Those arrows are pretty narrow, which means the rates are quite slow, which means in the tropics, fish are not moving along this depth gradient very readily. But when we look over in the temperate fishes, there's something else going on. Those arrows are much wider. They're basically bouncing around in this depth gradient way more. And specifically in the deeper depth categories, we're seeing a lot more transitions, both within the latitude and across to other latitudes. But polar latitudes are king. So these arrows are massive. This is where basically all the depth transitions globally, what we see across fishes, are almost exclusively located in the poles. Basically, polar fish are able to take advantage of this depth gradient in a way fishes of any other latitude aren't able to. So what this means is that, number one, we're finding increased rates of depth evolution in these 5 focal clades. Although I really collapsed some very broad swaths of the ocean down into some very general categories for the purposes of this study, each depth zone itself encompasses a lot of habitat variation and opportunity to diversify, and these clades are able to take advantage of it. We also know that just more broadly, depth lability is spurring speciation across fishes. And this points to a more generalizable effect of depth on evolutionary dynamics at large. And then we also know that invasions of the deep sea are largely occurring at high latitudes. So why are high latitudes so darn special? What is going on there? And I'm going to drink water and make you think about that for a second. Man, it's hard on your voice to talk this long. Okay, so I think this relates back to the fact that Earth's conditions haven't been stable on a multitude of temporal and spatial scales. Number one, seasonality. So this relates back to some really fundamental ecological theory proposed by Dan Jansen in 1967, which is the idea that mountain passes are higher in the tropics. So this is just to say that because there's less seasonal variation in the tropics, species moving along an elevational gradient are going to be a lot more likely to encounter temperatures that they're maladapted for. Whereas in the temperate zones, there's a lot more seasonal variation, so they don't encounter those temperatures quite to the same degree. And although this theory was formulated around terrestrial systems and elevational gradients, I think analogous principles apply to the depth gradient in marine systems. So basically, diversification along the depth gradient, it might be comparatively easier for fishes that are already exposed to greater seasonal variation and climatic heterogeneity compared to tropical fishes. We also know that the Earth's conditions have not been stable on much larger timescales. So glaciation patterns are known to influence patterns of diversification, particularly in high latitude fishes. So we know that periods of glacial maxima are associated with the proliferation of a lot of these clades, like eelpouts, like Antarctic ice fishes. And during these periods of glacial maxima, we know that there's massive extinction events at the poles. And it's thought that species are kind of relocated to a few regions of open ocean refugia during these time periods. And then during warmer eras, they're able to recolonize these areas and proliferate and speciate like crazy. And then finally, just the normal stratification of the ocean. We already know that there's much less of a thermocline at the poles compared to the tropics, but it's not just temperature, it's a lot of other oceanographic parameters like oxygen availability and density. So in other words, the depth profiles of high latitude environments tend to just be a lot more uniform across multiple dimensions than in the tropics. And that just lowers those abiotic barriers to dispersal for high latitude organisms. So it's not quite this dichotomy of clade A versus clade B that I presented in the beginning. It's actually polar clades versus tropical clades here, where we know that polar clades are able to use their kind of clade-specific ecological factors and diversify along this depth gradient. They have the accessibility to do it, whereas tropical clades just don't enjoy that privilege. So this implies that depth evolution may actually be sustaining that latitudinal diversity gradient of speciation rates that I showed at the beginning. And I think more crucially, as our oceans are warming, what this means is these species are going to be facing some steeper barriers to dispersal along this depth gradient. And this means that over time, what we may see is a general slowdown of this engine of biodiversification. So I hope that through these 3 vignettes, what I've been able to demonstrate is how studying different aspects of diversity can bring us from this oversimplified AI perspective to a more realistic image of what diversity is like. And although I've only focused today on habitat and a couple of different aspects of diversity, we know that it's a much more complex system with a multitude of factors involved and that can shape patterns of diversification. And we really need more of an integrative macroevolutionary perspective to bridge these two, to bridge this gap. And I think finally what I want to leave you with is the emphasis that modern day diversity of fishes is a consequence of millions of years of evolution. And the better understanding we have of those factors involved allows us to better predict the future of this diversity. So with that, I would like to thank so many people involved in helping to collect data, particularly for those morphological measurements at the start, and I will happily take questions. Thank you.